Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this gluconeogenesis lecture, I am going to introduce you what is gluconeogenesis, and also I will go over timelines for various energy sources during fasting and starvation, and then briefly I will introduce you what are the sources of gluconeogenesis. Now let's get into the details. What is actually a gluconeogenesis is? Gluconeogenesis, it is a formation of glucose from a non-carbohydrate sources. Example, conversion of lactate into glucose molecule, conversion of amino acids like alanine into glucose molecule, conversion of an organic molecule like propionyl-CoA into glucose molecule, or conversion of glycerol into glucose molecule. So this is what is a gluconeogenesis is, and why gluconeogenesis is needed for us. Gluconeogenesis is a process of formation of glucose under fasting condition and starvation to provide energy needs of our cells, especially red blood cell and the brain. And by doing this gluconeogenesis, the energy needs of cells is supported until a food source is taken in the form of diet. Now, where exactly gluconeogenesis is going on? Gluconeogenesis predominantly will be going on in the liver. Liver is the predominant organ where gluconeogenesis will be going on. Other than liver, to a certain extent, it will be going on in the kidney, especially in the cortex of kidney. This is under extreme starvation condition. Cortex of kidney can synthesize glucose by conducting gluconeogenesis, and the glucose that is produced by the cortex of kidney will be simply used by the medulla there. So that means kidney do not contribute to blood glucose level, unlike a liver that gives glucose into the blood. Gluconeogenesis conducted in the liver is only for other tissues because it will be secreted into the bloodstream. Whereas the gluconeogenesis in the kidney, it is only for its own use for medulla. And also to a certain extent, small intestine will also synthesize glucose by gluconeogenesis, but it generally do not contribute it to blood glucose level. Now, what are the major precursors for gluconeogenesis? As I said earlier, major precursors for gluconeogenesis is alanine coming from skeletal muscle, alanine. It will be lactate coming from anaerobic glycolysis. It will be glycerol that is coming from adipose tissue triacyl glycerol breakdown, or it will be a propionyl-CoA that come from oxidation of certain biomolecules in our body. Now, as I said earlier, liver is the major organ where gluconeogenesis is going on, and during starvation, kidney can also to certain extent conduct gluconeogenesis. Small intestine also express key gluconeogenic enzymes, but its contribution to blood glucose is not significant or not clear. Now it is important to understand the timeline for energy sources during fasting and starvation. It is because that will give us an understanding how our resources present in our body will be undergoing a metabolic modulation type of thing to provide energy source for brain, for red blood cell, for skeletal muscle, for heart, so that person will survive until the food arrives. So let's get into the timeline here. So the y-axis, it is indicating plasma levels of molecules. Now the first thing is, as you see, glucose. And the top line that you are seeing here, glucose, it is slowly, it will fall, the levels of glucose will slowly fall as we go from few hours of fasting towards first week of fasting. And as we move from first week of fasting all the way, so there will be constant amount of glucose that is maintained here, which is about 80 milligram per deciliter. That's the lower fasting level of glucose. Now, what are the processes that will maintain this lower fasting level of glucose? A couple of hours after fasting, like usually 2 to 3 hours after meals, and then moving on to first week and then afterwards. So the very first process that will contribute to blood glucose level is glycogenolysis. As you see here, the glycogenolysis is the breakdown of glycogen, and especially in the liver. And 2 to 3 hours after meals, a glycogen in the liver is broken down into glucose 1-phosphate, and glucose 1-phosphate further converted into glucose 6-phosphate, and that glucose 6-phosphate will be converted to glucose and released into the blood. Now the glycogen degradation, that is glycogenolysis, starts 2 to 3 hours after meals, and then it continues to rise until 12 hours of fasting, where it peaks, and from the peak it will start to fall down for next 12 hours. That means by 24 hours there will be complete degradation of glycogen. So glycogen contribution to blood glucose level maintenance during post-absorptive period and fasting state is maximum at 12 hours and then continue to decline. Now as the blood glucose contribution from glycogenolysis declines, so gluconeogenesis starts. Now the gluconeogenesis starts 2 to 3 hours in the post-absorptive period, but it's at a very baseline. By 12 hours it is steaming up. By 24 hours gluconeogenesis will be at peak, and it will be at peak for 1 full week, and then slowly it drops to certain range and it will be maintained in that particular range. That's what is the timeline for gluconeogenesis. That means gluconeogenesis, it peaks by 24 hours. After 1 week it is slowly fall down to certain range, and then it is continued to maintain in that range. Now as these changes are going on, so during post-absorptive period going into fasting state, so fatty acids from the adipose tissues are flooding into the blood, and these fatty acids, they slowly rise in the blood. By the end of 1 week, there will be significant amount of fatty acids there. After that, they continue to rise for 2 to 3 weeks, and then they will take that peak stage there and maintain it there. Now as the fatty acid levels are continuously rising, fatty acids which are undergoing oxidation in peripheral tissues, including in the liver, and in the liver fatty acid oxidation which is giving acetyl-CoA, and acetyl-CoA, it will be converted into ketone body in the liver, and ketone bodies will start to appear in the blood little more than 12 hours, but not in a significant quantities. But by 24 hours after fasting, there will be significant appearance of ketone bodies, and for sure by 72 hours, that is 3 days, there will be more ketone bodies appearing in the blood. And by this time our brain is start to use ketone bodies, although not adapted itself to use ketone body, it starts to use ketone body, so that it takes 2 to 3 weeks from that point to completely adapt to use ketone body and decrease its use of glucose. And that's how ketone bodies are used by the brain and also other organs. And the glucose is spared by the brain for the red blood cell. Likewise, fatty acids are used by the skeletal muscle and spare ketone bodies for the brain. So like this, a metabolic adaptation, I would call it as metabolic adaptation, will be going on in between the tissues to use the energy resources during fasting and starvation. So just to recap, blood glucose level is maintained at lower range of fasting, that is between 70 to 80 milligrams per deciliter. Glycogenolysis peaks at 12 hours, and the stores are completely depleted by 24 hours. Gluconeogenesis peaks by 24 hours and it will be there for another 4 to 5 days and then drops to certain level and maintained there. Fatty acids appear in the blood by 3 to 4 hours after fasting, keep rising for 2 to 3 weeks, and ketone bodies significantly appear in the blood for 24 to 48 hours and continue to rise for next 2 to 3 weeks. And this is the timeline for various energy sources during fasting and starvation. Now let's move on to see what are the precursors for gluconeogenesis. So the major carbon sources for gluconeogenesis in humans is glucogenic amino acids. You know, glucogenic amino acids are out of 20 standard amino acids, we have 18 of them. 18 amino acids are glucogenic because 18 amino acids can be, whenever these amino acids are undergoing catabolism, their carbon skeleton in one or the other way can be converted to glucose. Now how to remember those 18 glucogenic amino acids? All you need to do is to remember those 2 amino acids which are ketogenic, because we have only 20 amino acids, 18 of them are glucogenic, means 2 of them are ketogenic. So 2 ketogenic amino acids are leucine and lysine. Leucine and lysine, these are the 2 ketogenic amino acids. How do you remember leucine and lysine? Mnemonic is Lucy-Lyse. Okay, that's the mnemonic. So that means if you remember leucine and lysine, so other than leucine and lysine, all other amino acids are glucogenic. So all of them in one or the other way will contribute to glucose formation in gluconeogenesis process. That's the one of the source of gluconeogenesis. Second is lactate coming from anaerobic glycolysis, especially from the red blood cell or any tissues where anaerobic glycolysis is going on. Like example, in the skin, in the retina of the eyes, cornea, lens, right, adrenal medulla. So things like this where anaerobic glycolysis is going on to a certain extent. So lactate coming from there can be used for gluconeogenesis. Glycerol coming from adipose tissue lipolysis can be converted into glucose in the liver. Propionyl-CoA coming from various sources, which you can remember the sources of propionyl-CoA by using a mnemonic Bob needs vitamin M. And what is there? What actually is the Bob needs vitamin M? I already explained you in my previous video. Let me tell you again. B for bile acid synthesis. Bile acid synthesis gives propionyl-CoA as the one of the product. O for oxidation of odd number carbon fatty acid. Oxidation of odd number carbon fatty acid. O for O is odd. Odd is the O there in Bob. And another B is for oxidation of branched chain fatty acid. Branched chain fatty acid oxidation. So we can write it like that instead of writing oxidation of odd number carbon fatty acid. So you can write it as odd number carbon fatty acid oxidation, so that you remember what is O there. And the other is branched chain fatty acid oxidation, that is another B. V for oxidation of valine oxidation. Other is I for isoleucine oxidation, and T for threonine oxidation, and M for methionine oxidation. So oxidation of valine, isoleucine, threonine and methionine, all of them will give propionyl-CoA. So like this, there are different sources of propionyl-CoA, and that propionyl-CoA can go into gluconeogenesis. So that's all about our timeline for gluconeogenesis, introduction to gluconeogenesis, and the major precursors for gluconeogenesis process. Thank you.